Chapter 3: Evolution and Diversity of Green and Land Plants. The green plants. The green plants, or Chlorobionta, are a monophyletic group of eukaryotic organisms that includes what have traditionally been called green algae, plus the land plants or embryophytes, figure 3.1. Like all eukaryotes, the Chlorobionta have cells with membrane-bound organelles, including a nucleus containing chromosomes composed of linear chains of DNA bound to proteins that are sorted during cell division by mitosis, microtubules, mitochondria, and endoplasmic reticulum, vesicles, and Golgi bodies. Although the interrelationships of non-land plants Chlorobionta will not be covered in detail here, it is important to realize that some of the evolutionary innovations or apomorphisms that were normally associated with land plants actually arose before plants colonized the land. Several apomorphisms unite the Chlorobionta, figure 3.1. One possible novelty of this group is cellulosic cell wall, figure 3.2a. Cellulose, like starch, is a polysaccharide, but one in which the glucose sugar units are bounded in beta 1 to 4 position. The slight change in chemical bound position results in a very different molecule. Cellulose is secreted outside the plasma membrane as microscopic fiber-like units called microfibrils that are further intertwined into larger fibril units, forming a supportive meshwork. The function of cellulose is to impart rigidity to the cell, acting as a sort of cellular exoskeleton. The evolution of a cellulosic cell wall was a preamble to the further evolution of more complex types of growth, particularly of self-supporting shoot system. It is not clear if a cellulosic cell wall constitutes an apomorphy for a Chlorobionta alone, as it may have evolved much earlier, consulting an apomorphy for. For the Chlorobionta plus one or more other groups. In any case, its adaptive significance seems clear. Perhaps, the primary apomorphy for Chlorobionta is a specialized type of chloroplast. As discussed in Chapter 1, chloroplasts are one of the major defining characteristics of traditionally defined plants. Their adaptive significance as organelles functioning in photosynthesis, the conversion of light energy to chemical energy, is unique. Chloroplasts in the Chlorobionta, the green plants, differ from those of most other organisms, such as the red and brown algae. In number 1, containing chlorophyll B in addition to chlorophyll A, the former of which acts as a necessary pigment in light capture. Having thylakoids, the chlorophyll-containing membranes that are stacked into a grana, which are pancake-like aggregations. Manufacturing as a storage product through starch a polymer of glucose sugar units, in which the glucose molecules are chemically bounded in an alpha 1,4 position. Thus, all green plants from filamentous green algae in a pond or tide pool to giant sequoia or eucalyptus trees have this same type of chloroplast. Recent data imply that the chloroplasts found in the green algae plants today were modified from those that evolved via endosymbiosis, the intracellular cohabitation of an independently living unicellular prokaryote inside eukaryote cells. The Chlorobionta as a whole are classified as two sister groups, chlorophytes or chlorophyceae and stereotypes or stereotophyceae. The traditional green algae are a paraphyletic group. Why the name is placed in quotation mark and are defined as the primarily aquatic Chlorobionta, consisting of all chlorophytes and the non-land plant stereotophytes. Green algae occur in a tremendous variety of morphological forms. These include flagellated unicells, with or without flagella, thalloid forms, motile and non-motile colonies, the non-motile filaments. Many have flagellated motile cells at least one phase of their life history. Green algae inhabit fresh and marine matters, and some live in or soil, or other terrestrial but moist habitats. The primitive type of green plant sexual reproduction seems to have been the production of flagellate haploid gametes that are isomorphic, that is, the look identical. Fertilization occurs by union of two these gametes resulting in a diploid zygote. The zygote, which is a free living, then divides in meiosis to form 4 haploid spores, each of which may germinate and develop into a new haploid individual which produces more gametes, completing what is termed a haplontic life cycle. Within the stretophyte lineage that gave rise to the land plants, a new innovation evolved that may have been pre-adaptations to a survival on land. First of this was the evolution of oogamy, a type of sexual reproduction in which one gamete, the egg, becomes larger and non-flagellate. The other gamete is by default called a sperm cell. Oogamy is found in all land plants and independently evolved in many other groups, including many other algae in the animals. Two other evolutionary novelties that occurred prior to the evolution of land plants were retention of the egg and retention of the zygote on the parent body. Retention of the egg and zygote was adaptive, at least in part, by making possible the further nutritional dependence of the zygote upon the haploid plant, ultimately leading to the saprophyte. Several other amorphisms of and within the Chlorobionta include ultrastructural and specialization of flagella and some feature of biochemistry. Although these have been valuable in elucidating polygenetic relationships, their adaptive significance is unclear, and they will not be considered further here. An apomorphy for the charophytes, a clade within the stereophytes that includes the coleochate Charles and the land plants, a plasmodesmata. Plasmodesmata are essentially pores in the primary cell wall through which membrane traversed between cells, allowing for transfer of compounds between cells. Plasmodesmata may function in more efficient or rapid transport of solutes, including regulatory and growth-mediating compounds such as hormones. Members of Eukaryotes, such as genera Chara and Nitiella, or perhaps the closest living relatives to the land plants. These aquatic organisms form whorls of lateral branches and grow by means of a single apical cell, resembling that some land plants, the eukaryotes. The diversity of non-vascular land plants. During the early evolution of land plants, these major monophyletic lineages diverged before the vascular plants. These lineages